Good evening everyone. So in continuation to the last session that we had, the topic for today is transpiration and translocation. Okay, so this is the topic for today. So just to give you a brief intro as to what we just discussed in the last session, because this is a continuation of what we already discussed. Yeah, so we've seen how the water is absorbed from the root hair, and then how the water travels all the way to crossing the Caspian strips of the endodermis into the xylem, which is sitting at the center. Okay, so we talked about the apoplastic pathway and the symplastic pathway. Both pathways finally get the water here, and along with the water the ions are being transferred. Okay, along with the water ions are being transferred. So as a result of which the ion concentration inside the xylem is increasing. Okay, and whenever the solute concentration increases, the water potential decreases. So the moment water reaches the xylem, more and more ions are coming along with the water, so the water potential here is going down. So here in the water potential of soil water is still high, because there will be very less ions dissolved in the soil when compared to the concentrated ions here. Okay, ions are getting accumulated, because of which concentration is happening. So the water potential here is high, the water potential here is low, because of which the water is continuously drawn from the outside soil to the xylem. Okay, so today's session we'll discuss how the water is actually transported from the xylem. Okay, this is in the root, so to the various parts of the plant against the gravity. Okay, that's one thing. Second thing, how the food is actually transported. Food is made by the leaves, we all know that. How is the food which is made in the leaves transported all the way to the various parts of the plant? Okay, and there are some variations to that, and we'll discuss even that. Okay, so those are the two things that we'll discuss today. So we'll start with a brief discussion about the vascular bundles, because these two processes are because of the vascular bundles. Okay, vascular bundles are nothing but your xylem and phloem. Okay, so these two are your vascular bundles. We'll start brief description about what are these and what type of cells are these made up of. Why is that significant? Because unless until you understand how they are arranged, it becomes difficult for you to visualize the movement in a plant. Okay, the movement of water or food in a plant. Okay, so let's just understand them clearly, how the structure is, how they are arranged in a plant, and then it becomes clear for us as to how the water is actually moving between these cells and how the food is actually moving between one cell to another cell. Okay, so we'll first understand that. Then we'll talk about ascent of xylem sap, that is movement of water in the upward direction, okay, against the gravity through xylem. Okay, so that's the next topic which we'll discuss. So as a part of this, there are three theories which are put forward to explain how the water is actually moving against the gravity and reaching such heights. Okay, the three theories are the root pressure theory. I introduced the term root pressure in the last session. We'll discuss the root pressure theory. Then the capillarity theory, right? The capillarity theory. Then we have the final thing, which is cohesion tension theory or cohesion theory. Simply it is also called as cohesion theory, cohesion tension theory or cohesion theory. So these are the three things which we'll discuss as a part of how the water actually moves from the roots to the apex of the plant. Okay, पूरा टॉप तक पानी कैसे जाएगा वो हम डिस्कस करेंगे. ये तीन theories, these people tried to explain how the water actually moves from the roots all the way to the apex or the top of the plant. Okay, the top region of the plant. Then once we discuss that, okay, we'll discuss transpiration, which is nothing but evaporation or loss of water from the plants in the form of water vapor. Okay, and then we'll look at the translocation of minerals, that is how the minerals are actually moved from one region to another. And finally we'll look at the translocation of the sugars, sugars made in the leaves to various parts of the plants. Okay, so these are the things which we'll cover today. Okay, so let's start with the first thing, which is xylem. Okay, now if you look at xylem, the xylem is actually made up of three kinds of cells. Okay, three kinds of components. The fibers, we have the fibers, then we have the parenchyma cells, right, और the तीसरा चीज जो है वो है ट्रैकियरी एलिमेंट्स. ये तीनों चीजों से xylem बनता है. xylem tissue जो है, xylem tissue is actually made up of these three things. Okay, when I say trachyary elements are mainly of you have two types of trachyary elements. One is the tracheids, okay, and the other one you have tracheids, and the other one is your you have tracheids and just give me a second. Yeah, so the other one is your vessel elements. Sorry, completely blacked out for a minute. Okay, so you have tracheids and vessel elements. Okay, so these are the two things which are, so we'll look at the fibers. Fibers are basically there for structural support. Okay, they don't do anything, they just give structural support. Okay, they have a lot of strength and they give structural support. Then you have parenchymal cells, and these parenchymal cells usually are involved in storage. Some of these parenchymal cells actually store materials, okay, in the xylem tissue. Then you have trachyary elements. These are the ones which are actually responsible for the transport or the transfer of water from the roots all the way to the tip of the plant. Okay, so now between these two, if you take tracheids and vessel elements, both of these have a few things in common. Okay, both of them have a few things in common. Both of them are made up of elongated cells, okay, and they both have secondary cell walls, right? They both have secondary cell walls. What else? And the most important prominent feature of xylem tissue is they don't have any protoplasm. Okay, protoplasm is nothing but the contents which are present inside the plasma membrane. All the contents inside the plasma membrane together we call it protoplasm, and these cells lack protoplasm. So the basic components, cytoplasm or the nucleus, nothing is there. These are like dead shells. Okay, the outer layers are present, but the inner contents are gone. So these are dead cells. Okay, they are made up of dead cells, and because the inner contents are gone, they are hollow. Okay, they are hollow, and these hollow cells are interconnected. These hollow cells are interconnected. I'm talking about tracheids here. Okay, and these are the common things between your tracheids and vessel elements. Okay, common things between tracheids and vessel elements. They have dead cells. Okay, and these dead cells are interconnected. Okay, so let's look at tracheid cells first. Yeah, so this is your tracheid cell. Okay, so this is a dead cell and it is hollow. Inside there's nothing there, and if you can see, there are many holes on its surface, pits. Okay, there are many pits on its surface. Okay, now these tracheids are actually interconnected. So when I say interconnected, okay, there might be another tracheid here. Okay, right next to this there might be another tracheid, and it has pores, and through these pores there is a connection. Okay, let me just show you. Through the pores they have a connection. So you can see here, you see how through the pores they are actually connected. Okay, they are connected through the pores, and not just there, even towards the ends. So this is complete one tracheid. Okay, this is a tracheid. This is one complete tracheid, and this complete tracheid is connected towards the end also. Okay, usually they are connected well from end to end. So you have one tracheid here, it will end and it is connected to the next tracheid, it is connected to the next tracheid. So the entire thing is interconnected. The tracheids are interconnected, not just vertically, not just in this direction, side by side also they are connected, because the pores are not everywhere, wherever there are pores aligned. So you see here the pores are not aligned, but here you see the pores are aligned. Okay, wherever the pores are close by and they are aligned perfectly, there the exchange of material can happen from one tracheid to other tracheid, like this also. So from here it can move like this. Okay, let me show you another image. Yeah, you see the tracheids, this is one tracheid and this is another tracheid, which are connected by, the pores are exactly aligned here, the end to end connection is there, and the water can simply pass from this tracheid, okay, through the pores into the next tracheid, and through the pores into the next tracheid. So this is how the entire thing is interconnected. The entire thing is interconnected, and these are hollow cells. Okay, these are hollow cells. Okay, now vessels are also hollow. They are a bit bigger than your tracheids. They are a bit bigger than your tracheids. Now when I say the pores are actually aligned, okay, you have one pore, okay, and right next to it you have the second pore. So the pores are actually aligned. You see here, you see in this particular thing, the two pores are side by side. Now this is what we call a pore pair. Okay, it's a pore pair. Okay, so you have pore pairs through which water can move from one tracheid of the xylem tissue to the next tracheid. Okay, now these pore pairs can be bordered or maybe without border. Okay, they can be bordered or they might not have any border. If there is border, the border is because of the deposition of secondary tissue. Okay, so whenever the secondary tissue forms, the secondary tissue gets deposited around the border, okay, around the pore, and that is what makes it a bordered pore. Okay, if it is a bordered pore or bordered pit, okay, pore pair or pit pair. Okay, if it's a pore pair, or it's also called as pit. Okay, I can see pits. Okay, so the bordered pits have secondary deposits on it, because of which you will see that they look something like this. Okay, they look something like this. So this is, yeah, you can see here, this is one tracheary element. The entire thing is one tracheary element, right? And this is the next tracheary element, and the two pores are aligned here, right? And this pit doesn't have any border, and this pit is called the simple pit. A pit without a border is called a simple pit. However, some pits have a border. You see here, thickened border, right? And in these thickened borders, okay, in these thickened borders, you have a thickened central portion, okay, a thickened central portion, okay, and this portion is called torus. Okay, let me just quickly tell you again, just quickly. So you have tracheate elements, and these are interconnected, okay, end to end as well as side by side. End to end as well as side by side, they are connected. They are connected through pits. Okay, now these pits can be bordered, or they can be without any border. If there is no border, then it is called a simple pit. If it is bordered, okay, along with the border you will see a central enlarged portion, central thick portion, and that thick portion is called the torus. And torus is something which helps with the pressure control on either sides of the cells. Okay, pressure control on either sides of the cells. You just have to learn the term, okay, at your level. If you understand the term, that's enough. Okay, the pits between the tracheates can be bordered, or they can be without any border. If there is no border, it is called the simple pit, and if it is bordered, then in the central portion you will see a thick region, and that thick region is what we call the torus. Okay, now these tracheates are generally, okay, 10 to 50 micrometer in diameter. Okay, 10 to 50 micrometer in diameter, and maximum length that you can see is 1 to 3 centimeters. Okay, the length will be 1 to 3 centimeters, and the diameter will be 10 to 50 micrometer. Very, very small. Diameter is very, very small. You will be able to see the cells because the length is close to 1 to 3 centimeters, somewhere of this size. Okay, so yeah, this is about the tracheates. And another important thing that you have to understand here is the tracheates. Okay, the tracheates have lignin deposition. Okay, in addition to the cellulose and hemicellulose, which forms the cell walls of these cells, okay, they have lignin deposited in their walls. In their walls they have lignin deposition. Okay, and because of that lignin deposition, they become impermeable to water. Impermeable, that is, water will not diffuse through the walls. Okay, they become like plastic pipes. Okay, the water will not diffuse through the walls. However, they can diffuse through the pits. The water can still move through the pits which are there in the tracheates, but not through the walls. Okay, usually in live cells you can see the water actually moving from outside to inside through the walls, or from inside to outside through the walls. Okay, so but in these cells you won't see that because they are deposited with lignin, and these are dead cells, hollow cells, and if the water has to pass, it has to pass through the pits which are there on these elements, okay, on these tracheates. Now the second thing is your vessels. Okay, now the vessels are more or less like the tracheate elements. However, the vessels are a bit bigger. They have diameters close to, yeah, they have a bigger diameter than these, and their length varies actually. It can vary from 40 to 60 micrometer in some plants, and it can go all the way to 10 meters in plants like oak. This one thing can be 10 meters. Okay, so Amitabh Bachchan is 1.9 or 1.8 meters. Okay, so this thing can go all the way to 10 meters. So they can be such long vessels inside the xylem. Okay, inside the xylem tissue of some plants like oak, such big vessels can be found. Okay, and these vessels are different from the tracheids. They have perforated ends here. Okay, they have perforated ends, and they connect with each other through these perforated ends. So you have one vessel here, the next vessel will attach here, and through these perforated things the materials are passed on. And just like the tracheid elements, even these have pits, and just like the tracheids, water will pass laterally side to side or from top to bottom. Okay, so that way these are not different. The only major difference between these two is the diameter. Okay, these are a bit bigger than these, and the other difference is tracheids are found only in the gymnosperms. Okay, in the gymnosperms you will find only tracheids. Okay, the water transport is by the xylem in the gymnosperms, but the xylem is entirely made up of tracheids in the gymnosperms. However, in the case of angiosperms, okay, in addition to the tracheids will be there, but majority will be your vessels. The vessels will be the major ones which are involved in the transport of water. In addition to the vessels, you will actually see the tracheids as well. Okay, you will find the tracheids as well. So just to summarize, you have in the xylem, right? In the xylem, you have three kinds of cells, right? The fibers, the parenchyma cells, and the tracheary elements. The tracheary elements, right, are the ones which are actually involved in water transport, and the tracheary elements can be of two types. One is your tracheids, right, and the other one is your vessels, right. Tracheids are found only in the gymnosperms you will see only tracheids. However, in angiosperms you will see both tracheids as well as vessels. Okay, now they both connect end to end. Okay, they have pores all over their body, and they both connect end to end. Okay, they both connect laterally also through pit pores, okay, or pits, and these pits can be bordered or they can be without any border. If there is border, you just have to remember one term, that the bordered pits have a central bulged region called the torus, and that torus is involved in control of pressure on either sides of in the adjacent cells. Okay, you just have to remember torus, that's more than enough. Okay, now so with this information in mind, we'll move to the next part. Okay, by the way, vessels are considered to be more advanced than tracheids because they are found in the angiosperms. Gymnosperms are in terms of evolution. Gymnosperms are considered to be primitive when compared to your angiosperms. So tracheids being found only, or in gymnosperms only tracheids are found, that makes them less advanced than the vessels which are found in the angiosperms. Okay, now so the vessels have another advantage. They are superior than tracheids because they conduct the water much better than the tracheids. Okay, they conduct the water much better than the tracheids. That way also they are superior than the tracheids. Okay, now the next part, now that we have some basic understanding as to how the xylem tissue is and how it is actually arranged end to end, okay, to form a connection. So the entire plant from top to bottom will have the connection. Okay, the entire plant from top to bottom will have the connection. So just to come back to our next topic, which is ascent of sap, that is ascent of water, that is movement of water from the roots all the way to the top. You see that the water is actually coming from, so you have your root hairs, okay, through the root hairs the water is actually coming in, entering the epidermis, and by the apoplastic or the symplastic pathway, okay, symplast or the apoplast pathway, the water is reaching all the way to the xylem, okay. And when it reaches the xylem, I told you xylem is actually made up of many cells which are connected end to end, and they have pores on the sides as well as at the ends. In the ends the pores are aligned so that the water will move from one xylem element to the next through the pores directly, and the water can also move laterally like this, okay, because the pores are interconnected, okay, the pores are interconnected. So the entire xylem tissue is aligned here. Now what we are about to discuss is the water which is accumulated here, how is it actually moving from the xylem or through the xylem tissue to the various parts of the plant, okay. Now this is explained, like I said, movement of water from roots to the apex, that is the tip of the plant. How is this water actually moving? Okay. Now, in order to explain this, for instance, I'll tell you something. If you take sequoia plant, right? This sequoia plant is 110 meters tall, right? This is 110 meters tall. So the water from the ground, okay, from the ground has to go all the way to 110 meters, okay, before it is delivered to the leaves there, okay, before it is delivered to the leaves there. So how is it actually moving against the gravity and reaching the various parts of the plant? That is what we are about to discuss. Okay, now if you take a simple thing, okay, if you take a simple thing, I'll just give you simple, see this. So if you can actually create a big beaker, okay, which is let's say 20 meter tall or something, okay, very big beaker, and then a very big tube which is let's say 15 meters tall, and then put this tube into this beaker, okay, because of the, so your atmosphere has some pressure, okay, the atmospheric pressure is 101.3 kilopascal, okay, which is one atmospheric pressure, 1 bar, okay. So the water, because of this atmospheric pressure, will start to move up, okay, not just atmospheric pressure, the other factors we'll discuss that in a minute. So if you can create something like this, the maximum that the water can be pushed is to a height of 10.3 meters, okay. So people have calculated that if even, so this is the best case scenario. The best case scenario, if we can generate pressure, that pressure can push the water up to 10.3 meters. And in the example that we just discussed, I told you that some plants are as tall as 110 meters. So this pressure, this atmospheric pressure, okay, is nowhere near, okay, is nowhere sufficient to generate the pressures required to push the water all the way to the top regions of the plant, okay. So people always wondered how this is happening, how is the atmospheric pressure is not helping it, okay, to a large extent. So how is this water actually moving, okay, even if you put lots and lots of water to the plant, the pressure created there will not be sufficient to push the water up the plant. Okay, that's what we're trying to say here. So what is actually helping? There are three theories which have been proposed to explain how the water actually moves, okay, from the bottom to the top. The first one is the root pressure theory. Okay, then you have the capillarity theory. Then you have the cohesion tension theory. Okay, so these are the three theories which were proposed to explain how the water is actually moving to such enormous heights. Okay, so we'll start with the first thing, which is your root pressure theory. Okay, we'll start with the root pressure theory. Right, we already discussed the root pressure theory. Okay, in the last session we already discussed the root pressure theory, where I told you okay, so this is your xylem, cambium and phloem. So here you have your outer layer is your epidermis, then you have your cortex, and then in the center you have your xylem, and so this is your xylem and phloem. So this is your xylem, yeah, so this is your xylem and this portion is your phloem. Okay, now according to the root pressure theory, okay, according to the root pressure theory, we already know that the water will enter through the upperplast and the symplast pathway. Okay, the water will enter, so from, let's say here you have the root hair, the water is entering and it will through the upperplast and the symplast pathway, it will reach the xylem, that is this portion. Okay, it reaches the xylem. Once it reaches here, the water, okay, now you have your water, along with the water you are getting many ions. Okay, along with the water you are getting many ions. Now the ions, okay, which are accumulating here will lower the water potential, and because the water potential is going down, okay, because the water potential is going down, more and more water is pulled from outside into the xylem. So more and more water starts to pack here. Okay, more and more water starts to accumulate here. When that happens, the hydrostatic pressure here increases. Okay, the hydrostatic pressure here increases. Okay, when the hydrostatic pressure increases, the water is pushed upwards. The pressure up is low, but here it is high, so the water is pushed upwards. Okay, but what scientists found out is, you need at least, okay, so for that example that we discussed, for let's say 110 meter plant, okay, if you have to push the water all the way to the top, you need close to 2 to 3 megapascal pressure. Okay, a pressure of 2 to 3 megapascal. Okay, and the root pressure, okay, what the root pressure generates is just 0.1 to 0.5 megapascal. Okay, 0.1 to 0.5 megapascal, which is close to 15 or 16% of what we actually need. Okay, so root pressure can be explained, can be one of the reasons why the water is actually moving in the upward direction, but it cannot be explained in plants which are quite tall. Okay, now with this pressure, okay, maybe to some extent the water can be pushed in the upward direction. The water will be pushed in the upward direction, but this theory failed to explain, okay, the reason why the water is actually moving from the roots to the top of the plant even when there is no root pressure. Okay, in some plant species, there's no root pressure. Okay, the pressure which is generated as a result of water getting accumulated in the xylem. Okay, in some plants you don't see the root pressure. Even in those plants, the water is being transported all the way from the roots to the top. So root pressure theory will not explain that. Okay, so cannot explain that. So next theory that was put forward is the capillarity theory. Capillarity theory. Okay, now capillarity theory, we'll first understand what capillarity is. Okay, capillarity, or it is also called as capillary rise. Okay, so we'll first see what capillarity is. So if you take a beaker, okay, let's say the water level is till this point in that beaker, and if you introduce a capillary tube into this, okay, if you introduce a capillary tube into this, you will actually see the water level somewhere above the level of the water in the beaker. Okay, you will see the water level inside the capillary tube slightly above the level of the water inside the beaker. Okay, now why is this happening? Why is the water actually moving, okay, up inside the tube? Okay, so this movement of water up through the capillary tube is what we call the capillary rise or capillarity. You can simply call it capillarity. Now the capillarity or capillary rise will happen because of two reasons. Okay, and it happens because of the properties of the water. Capillary rise is because of the properties of the water. Okay, ये capillary rise जो है पानी के properties की वजह से होता है. And what are the two properties of water that actually makes or that are actually responsible for capillary rise? The two properties are adhesion and surface tension of water. Okay, and this surface tension is because of the cohesive forces. Okay, in some books you will see adhesion and cohesion. Okay, both are correct. Surface tension is because of the cohesive forces, so you can say adhesion and cohesion as well. Okay, both are correct. Now adhesion is basically, right, adhesion is basically interaction between water and walls of the tube. Okay, walls of the tube, right? So the walls have materials, okay, the walls have materials to which water gets attracted. Okay, so you have a tube like this, and let's say the water level is still here. Now what is happening? The molecules, water molecules here are getting attracted to the wall. Okay, so because of which they are getting attracted and slowly they are going up. Okay, so okay, we can understand that the molecules which are, you know, in touch with the wall are going up, right? Because they are getting attracted to the wall, right? They are getting attracted to some materials in the wall. But why is the entire, even here you will see water, why is the entire region moving up? Why is the entire thing moving up? The entire thing is moving up is because of the cohesive forces. Water has attraction not just towards the walls of the tube, water has interattractions. So water to water attraction is also very strong. Okay, one water molecule has a hydrogen bond with the other water molecules. So all the water molecules are interconnected with the hydrogen bonds, and because of this cohesive forces, what happens is one molecule is there, it is pulling, it is trying to attach to the wall, okay, and this molecule will successfully attach to the wall or bind to the wall, okay, or stick to the wall or adhere or gets it binds to the wall. Now in the process, what is it doing? It is actually pulling another water molecule which is bound to it, and this water molecule is pulling another water molecule which is bound to it. So this pulling is what is causing the water to actually rise. Okay, now how far will the water rise? Will it be indefinite? No, the water will rise till the pulling force till the pulling force is balanced by the gravity. Okay, when the two opposite forces are balanced, the water moving up will stop. Okay, the water will not move anymore because the water is now being pulled down by the gravity. So when these two opposing forces are balanced, that is when the water will stop, and till that point water will actually move up. Okay, and this moving up of water is what we call the capillarity rise. Okay, and capillary rise or capillarity. Okay, and now what people found is capillarity is responsible for pushing the water to a height of 0.75 meters. In plants, it can push the water up to a height of 0.75 meters. Okay, so in small plants, okay, capillarity could be one of the reasons why the water is actually going up. Okay, capillarity could be one of the reasons why water is actually going up. But again, capillarity theory cannot explain how the water is actually reaching a plant, okay, to the tip of the plant, which is almost 100 meters or 110 meters in height. Okay, in order to explain that concept, we have to look at the cohesion theory or cohesion tension theory. Okay, now this cohesion tension theory or cohesion theory was put forward by a scientist named H. Dixon. Okay, he is the one who actually proposed this theory. Okay, and this theory is based on one important assumption. Okay, this theory is based on one important assumption, and this is important. Okay, unless until this has been proved later, but when it was proposed, okay, जब propose किया था तब ये सोच के propose किया था कि there is a continuous column of water right, there's a continuous column of water from the tip of the roots through the stem to the mesophyll cells of the leaf. This was the main. So this, assuming this, okay, this theory was explained. Okay, this was, they said, okay, there is a continuous column of water from the, it was not proved at that point of time, but this assumption was made and this proposal was done. So what it essentially means is, if you take a plant, a big tall plant, and let's say these are the roots. Okay, inside the roots, like I said, all the xylem elements are connected to one another to form one long hollow interconnected tube. Okay, different xylem elements are connected one to one to form a long hollow tube. Okay, so through that tube the water will pass. Okay, through the tube the water will pass through the stem. Okay, the xylem is extending all the way through the stem to the leaves. How is it actually entering the leaves? In the leaves you have your veins. Okay, through the veins will have the vascular bundle, the xylem and the phloem. Okay, the veins will have the vascular bundle. So this xylem, which is starting all the way from the roots, is ending in the leaves which have the mesophyll cells. So this theory says, okay, this theory assumes that this entire thing is connected with a tube all the way from the tip of the root, okay, through the stem to the mesophyll cells in the leaf. Everything is connected with a big tube like thing, and this tube has actually a water column. Okay, there is water actually inside. Okay, water column, and everything is connected with this water column. So that's the assumption, everything is connected with this water column. That means it's like a straw. So you keep your straw in a glass and you start sucking from the top, right? So you will see actually that the water in the glass and the water reaching your mouth is one complete conduit or one complete connection. Okay, it's a simple connection. So that is what is continuous column. That is what is assumed. Okay, so there is continuous column of water between the tip of the roots and the mesophyll cells present in the leaf with the stem in between. Okay, so this is the first assumption which was made, and based on this assumption this theory was proposed. So here, so you see this is continuous. So this is the root tip, right? Root tip. From the root tip, that is root hair, the water is actually moving in, reaching the xylem, and through the xylem it is reaching all the way. The xylem is extending all the way into the leaf veins. Okay, leaf veins, and this is how the continuous column of water is forming. There is a continuous column of water starting from the root hair all the way to the mesophyll cells. These are the mesophyll cells of the leaf. Okay, there is a continuous column. Okay, now assuming this was proposed. So what is said is, so when the water reaches the leaf, okay, the water gets evaporated, which is nothing but your transpiration. Okay, the water gets evaporated. Now when the water gets evaporated, there is actually water surrounding these cells. Okay, the water is actually surrounding the cells. So you see here, so absorbed, and in the leaves these are the mesophyll cells. Okay, the green ones are your mesophyll cells. Surrounding the mesophyll cells you have your water which is coming from the xylem, which is being provided by the xylem. Now this water is getting evaporated. Okay, and I told you water has the adhesive as well as the cohesive property. Now because of this there's a tension created here. So slowly the water is drying up. Okay, slowly the water is drying up. Okay, it's like this. I'll explain it to you in the simple way possible. So you take a beaker, you can do this at home also. You take a beaker, okay, fill it up with water, any container, fill it up with water, put a sponge. Okay, you take any sponge, the porous sponge, right, and put a sponge in it. Okay, because of the capillary action the sponge will start to absorb water. Now if you switch on the air or apply some heat, whatever, the water from here will start to evaporate. The water from here, from the sponge, from the top portion of the sponge will start to evaporate. Now as the water evaporates, okay, the sponge becomes dry. When this becomes dry, it starts to pull in more and more water, and within no time you will actually see that this beaker, the level of water in the beaker will go down, and gradually the water will be completely absorbed by the sponge. Okay, that's exactly the same theory which is explained here. Okay, so because of this drying up, okay, because of the water vapor being released, right, a tension is created. Okay, a tension is created. Now because of the cohesive nature of the water, because the water molecules will not go alone, the water molecules here are getting dried up. So the water molecules, the little amount of water molecules which are left, they are trying to pull the water molecules which are behind them. Okay, now this water molecule here is trying to pull the water molecule behind it. So like that, the entire water molecules pulling, okay, each other is making sure that the tension is carried all the way to the root tips. So the water molecule here is pulling the water molecule behind it. Now because this is being pulled, this water molecule is being pulled by the water molecule, okay, which is in front of it. So the entire thing, because the water molecules, because of the cohesive nature of the water molecules, the entire thing, okay, will be pulled. So basically it's like putting a straw and then sucking it in. So you will continue to, okay, this is basically sucking in. So here it's not going into the mouth, it's simply going into the air, okay, and more water is being pulled, literally pulled from the roots to the various parts of the plant. Okay, so that is your cohesion tension theory. Because of the evaporation, tension is created, and because of the cohesive nature of the water will start pulling the water molecules behind them or adjacent to them, and this tension is carried all the way to the roots, and water is literally pulled from the roots through the plant all the way to the upper regions of the plant. Okay, so this was the theory which was proposed. Okay, and this theory, okay, this theory will work only when the cohesive forces of water, so the water molecule, okay, this water molecule is being pulled. So the water molecule, so let's say this water molecule is being pulled, right? Now this water molecule is pulling this, and this water molecule is pulling this, right? Like that, this water molecule is pulling this. So this will only work when these bonds are strong enough, right? Otherwise what will happen? It will simply break. Okay, so it's like this. You take a copper thread or any thread, okay, and try to pull it. Okay, if it breaks, it's not strong enough. It's not strong enough. And that strength is what we call the tensile strength. So only when you have good tensile strength, I'll give you an example. If you take a thread, normal cotton thread, and pull it, it will break. Okay, it will break because it has very less tensile strength. On the other hand, if you take a copper wire and try to pull it, it won't break that easily because it has higher tensile strength. It has higher tensile strength. And this will only work, this column of water, okay, this continuous column of water will only work if the tensile strength of water is good. Okay, if the tensile strength of water is good. And people started understanding the tensile strength of water, and they found that the tensile strength of water is minus 25 megapascal to minus 30 megapascal. That means the water can withstand a negative pressure of minus 25 megapascal to minus 30 megapascal. Okay, and what is the pressure that we need to push the water all the way from the bottom to the top? Okay, if you go back and check later, you will see that you need a pressure of 2 to 3 megapascal to push the water all the way from the bottom to the top of a plant which is 110 meters. So water has a lot more tensile strength than the pressure that is needed to push the water all the way to the top. In other words, the water will not break, the water stream will not break into two, okay, because it has very good tensile strength. It has very excellent. So this tensile strength, if you have to compare it with copper, it has 10% of the tensile strength of copper. Okay, the tensile strength of water is 10% of the tensile strength of, tensile strength of water is 10% of the tensile strength of copper. Okay, so water is that good, because of which the continuous stream is maintained, and because of this continuous stream, your evaporation will cause the negative, okay, tension, and this negative tension, because of the cohesive nature of the water, will make the water to pull the water molecule which is behind it, and this chain will follow, and this is carried all the way to the roots, and the water will be pulled literally from the soil into the roots through the plant all the way to the top. And this is the main reason why the water can actually reach such enormous heights. Okay, so this is the theory which is mainly accepted now, okay, because of which it is accepted that the water will reach to the top regions because of this thing. Okay, so that is about the three theories. Then the next aspect that I told we'll discuss is transpiration. Transpiration. Okay, now transpiration is nothing but loss of water from plants in the form of evaporation or water vapor. Loss of water as a result of evaporation from the plants. Okay, now if you see, the leaf has, like I said, the mesophyll cells, and these mesophyll cells have something called stomata. Okay, the mesophyll cells have openings called stomata. Okay, on the surface you have your stomata. The leaf structure has stomata on its surface. Okay, and these stomata are the openings through which not just water but the gases are also exchanged. Okay, the gases are also exchanged. Now this stomata, okay, so this is your xylem and phloem. You have your xylem and phloem, right? You have your mesophyll tissue. This is parasite mesophyll tissue. The entire thing is mesophyll tissue only. Okay, spongy mesophyll tissue. You have your upper epidermis, and in the epidermis you will actually see the stomata. Okay, you will actually see the stomata in the upper epidermis. Now the water is lost through these pores called the stomata. Okay, but these stomata are not always open. These pores are not always open. They are guarded. Okay, they are guarded by cells called guard cells. Okay, a pair of cells will be there on either side of this pore. Okay, the stomatal aperture is actually guarded by the guard cells. Okay, now how then, if they are being guarded, how is the water vapor actually going out, or the carbon dioxide and oxygen coming in? Okay, now again here what you have to understand here is, okay, so this is your stomatal opening, right? This is your stomatal opening which you will see in the epidermal layer. Okay, and now how is this actually being controlled? How is this actually closing and opening? Okay, that is something which we will see now. Okay, now you see these are your guard cells. This is one guard cell and this is the other guard cell. So you have two guard cells. Okay, I'm explaining the mechanism of transpiration. You have two guard cells, right? Now these two guard cells are actually protecting the central pore here. They are actually protecting the central pore. Now when the light, okay, in the presence of light, what happens here is, you have, these are nothing but cells, and cells have many proteins on their surface. You have active, okay, you have proteins which actively pump. When I say active, they are using energy. They use energy and pump protons outside from inside the cells. These are the two cells. This is one cell and this is one cell. So from inside the cell they pump protons, and in the process they pick up the potassium and the chloride ion. Okay, those protons are pumped out, and the potassium and chloride ion are taken in. So you have several ions getting accumulated here. Okay, and I told you already, the minute the solute particles or the ions concentration increases, what will happen? When the ion concentration increases, the water will start to move in. Okay, because ion concentration increases, then water potential decreases. And if you remember the last session, the water will always move from higher water potential to lower water potential. So the water will start now moving from outside into the guard cells. The water will start moving from outside into the guard cells. Okay, the water is actually moving in. Now, when the water starts to move in, okay, these cells bulge up. The turgor pressure will increase. The inside pressure of these cells will increase. And when these cells, when these cells, they're basically pushed towards the wall. They are designed in such a way that when they are pushed or when they bulge, the central opening is visible now. Okay, so they have microfibrils, okay, cellulose microfibrils, which also actually help in the process. Okay, so basically the water will seep in, because of which these cells basically gets bloated or bulged. Okay, so because initially it was hypertonic, now the water will start to move in, and now inside the cells it was hypertonic. So now what will happen? They bulge in and the pore is now revealed. The pore gets opened up. Okay, now this happens only in the presence of light. Okay, this happens, that is particularly in the morning. Okay, morning or during the daytime. Now what will happen during the night time? During the night time, when there is no light, okay, the potassium and the chloride ions will simply move through passive transport. The potassium and chloride ions will pass out of these guard cells through passive transport. No energy is used here. It's not active transport. They'll simply move out. When they move out, the water will also come out simply. Okay, when these cells lose water, obviously the entire thing will be closed again. Okay, the entire thing will be closed again. So this is how the transpiration is controlled. This is how the transpiration is controlled. When in the presence of light, okay, in the presence of light, the protons are actively pumped out by the guard cells, and in exchange they'll take up potassium and the chloride ion. Okay, this will facilitate the potassium and chloride ions to get in. When the potassium and chloride ions get in, the inner concentration of the guard cells, the inner ion concentration of the guard cells will increase, because of which the water potential will go down and the water will start to come in. When the water starts coming in, the guard cells bulge up, and this basically reveals or opens the stomatal aperture, the gap between. And during the night time, when there is no longer light, the potassium and chloride ions will simply diffuse through the guard cells, and now that the potassium and chloride ions are gone, the water potential inside is no longer high, so the water will simply diffuse through the water potential inside is no longer low, because of which the water will simply diffuse from the guard cells outside, and this causes the closure of the stomata. Okay, so that is about transpiration. Now transpiration, there are many things that are positive things because of transpiration. The first one is your it controls the temperature. Okay, it helps the plant to cool down, right? Then helps in the exchange of oxygen and carbon dioxide, right? And it is also responsible the evaporation that we just talked about in the cohesion tension theory. Okay, and because of which you are generating something called transpiration pull. Okay, because of the transpiration, the water is literally being pulled all the way from the roots to the top of the plant. That negative tension that we discussed there is nothing but the transpiration pull, is because of the transpiration process. Okay, and yeah, in addition to this, what else? The transpiration process also helps in it also helps in maintaining the water balance. Okay, so these are the various things which actually the positive aspects of transpiration. Okay, now we are already running out of time. I'll just quickly tell you about the translocation. Transpiration. Okay, first we'll talk about how the food is actually transported from, then I'll quickly wrap up by talking about the minerals thing. But first we'll talk about how the food is actually transported from the leaves from where it is made to the various parts of the plant. Okay, so here very quickly, since you already know the basics, so here this is your leaf, you have your mesophyll cells. Inside the mesophyll cells you have several chloroplast molecules, right? Inside the chloroplast molecules you have your thylakoid, okay, thylakoid membranes. Now these thylakoids are hollow button-like structures or hollow coin-like structures. Okay, now in these thylakoids, this is what happens. Okay, in these thylakoids, in the thylakoid membrane you have chlorophyll molecules. Okay, these green things are your chlorophyll molecules. This entire thing is your one thylakoid. Okay, this entire thing is your one coin structure, the thylakoid. Okay, in the thylakoid you have your in the membrane of the thylakoid you have several chlorophyll molecules. Okay, these chlorophyll molecules absorb the light, and then the light, because of which the energy which is obtained by them is transported to the reaction center. This reaction center will then split water into oxygen and protons. Okay, and the electrons are picked up, and these electrons are transported, and finally from the thylakoid membrane you are getting two things, okay, into the stroma. One is your NADPH and the other one is your ATP. These are two things which you are getting, and these two molecules are important, because of which the entire light dependent reaction happens. The entire light dependent reaction happens for getting two things, NADPH and ATP, the energy required for the subsequent steps. NADPH is the reducing equivalence. You need hydrogen, because ultimately from carbon dioxide you have to make, so you already have C and O, you have to make CHO, so you are getting your H from the NADPH. So you need these two things. So okay, from the light dependent reactions you are getting ATP, energy required for making your glucose or the sugar, that is sucrose, and you also need NADPH, the hydrogen from NADPH is required for the reduction of carbon dioxide. Okay, so using these two, inside the chloroplast your sugar is made. Okay, basically hexose phosphates are made, and from this the sugar which is made is your sucrose. In most plants it is the first component that is formed is your sucrose. Okay, now this sucrose, where is it actually forming? It is actually forming in the chloroplast. Okay, and inside the chloroplast you have sucrose, and where is this chloroplast present? The chloroplasts are actually present in the mesophyll cells. So inside the mesophyll cells of the leaf, have mesophyll cells have chloroplasts have thylakoids. In the thylakoids light dependent reaction will happen, generating NADPH and ATP. These two molecules are then used for the Kelvin cycle which happens in the stroma of the chloroplast, and because of which sucrose is formed. So the sucrose is now sitting in the leaves. Okay, the sucrose is now sitting in the leaves. Now let's see what happens to the sucrose which is sitting in the leaves. How is it actually transported to the various parts of the plant? Okay, so this is your mesophyll cell, the leaf cell, where your sucrose is formed. Now the sucrose from here will move into the companion cell. Companion cell is a part of phloem tissue. Okay, I told you about xylem tissue, the various components. I haven't discussed about phloem tissue since we didn't have time, but let me just tell you briefly. Phloem has two things. One is sieve elements or sieve tube cells, and the other one is your parenchyma tissue. Okay, and this parenchyma tissue is nothing but your in some regions you will see the parenchyma tissue closely associated with the phloem, and this is what we call the companion cells. Okay, I'm sorry I'm taking a lot of time today, but we need to finish this, we need to close this, so just bear with me for a few minutes. So you have your companion cells which are closely associated with your phloem, right? So this phloem is made up of, like I said, sieve tube cells or sieve elements, and just like your xylem tracheids and other things, they are also interconnected. Okay, and what you have here is what we call the sieve plates. Okay, the sieve tubes are connected by sieve plates, and these plates are porous, And one important difference between your xylem elements and the phloem elements or phloem components is that sieve tube cells are living cells. They are not dead cells like your tracheids or the vessels of your xylem. These are living cells. They are living cells. Okay, so the sucrose which is made in the leaf cell will now enter the companion cell, which is nothing but the parenchyma tissue of the phloem. Okay, and this is by simple passive transport. Okay, simple passive transport. Now once it enters the companion cell, from the companion cell into the sieve tube cell or sieve tube element, okay, active transport. It is actively transported, that is energy is used here, and the sucrose from this cell is pumped into the phloem sieve tube cell, okay, by active transport. Okay, now you have your sucrose getting accumulated in the sieve tube cell of the phloem. Okay, sieve tube cell of the phloem, sieve tube element of the phloem. Now once the solute starts to accumulate, what will happen? Again, same old story. If the solute starts to accumulate, water potential will go down. The water potential will go down. The minute water potential goes down, okay, what will happen? Here you have lower water potential. Adjacent to the phloem you have xylem with lot of water. So the water will start now moving from xylem. Like I said, these are the xylem elements have interconnected pores. Through those pores, the water will start moving from the xylem to the phloem. Okay, to the sieve tube elements of the phloem. Okay, now because of this, the hydrostatic pressure here will increase. The pressure here will increase. Okay, and because of this pressure, okay, so this, the entire thing, the phloem sap, okay, that is the phloem mixture, the liquid plus water plus sucrose here will start to move. Okay, will start to move because of the hydrostatic pressure generated there. Okay, you don't have such pressures here, so they start moving. Okay, when they reach regions or when they reach to the regions, so this region is called the source where it is actually made, and the regions where you need them, they are called the sink regions. Okay, it can be root, it can be some other component of the plant which is towards the which doesn't have leaves nearby. Okay, whatever it is, so it will be pushed. So from here again, from the sieve tube cell here, the sucrose is picked back again by the companion cell. Okay, and from the companion cell, okay, this is again active transport. The sucrose will simply diffuse to the sink cells. It can be any cell. Okay, if you're talking about a simple plant, it can be a root cell. Okay, so this is what we call the pressure flow hypothesis or pressure flow theory. So this is the theory by which, this is the theory which explains how the food which is actually made in the leaves is transported all the way to the root. Okay, all the way to the root. Now what you have to understand here is, some plants like your potato and all, what they do, they send them down, no problem, one direction. They, in the roots, they actually store the food. They actually store the food. So you have cells, so the food is being stored here. What happens in the, when the season changes, this food, the purpose why they are storing is, when the season changes, when there are unfavorable conditions, they can start using the stored food for their growth. That's the basic idea. So they are no longer making the food here. Now what should happen? The stored food here is broken down and in the reverse direction is sent to the various regions of the plant. Okay, is sent to the various regions of the plant. So unlike water which is unidirectional, unlike water which moves only in one direction through the xylem, which is from bottom to top, the flow of food, the movement of food, the movement of nutrients through phloem is bidirectional. It can happen from the leaves to the roots, or if the food is stored in the roots, from the roots all the way to the various parts of the plant. Okay, so this is the theory which actually supports, or which actually explains how the food is actually transported from the source to the sink. The source can be different things. If the season is good for making food, then they'll make food, store them. then the source becomes the leaves. But if the conditions are unfavorable, then the source becomes the roots or other regions which actually stored the food, and from there the food is actually moved to the various parts of the plant. Okay, so that is it. We are done with the various, we already discussed the various mechanisms by which the food is transported, how the water or food is moving through the various plants and all. So we are done with the transport of materials in plants. That chapter is done. Okay, in the next session we'll discuss the skeletal system. We'll briefly discuss the various aspects of skeletal system, and in detail we'll discuss the appendicular skeletal system. Okay, so some question was asked, what is protoplasm? Yeah, you can please send in your questions, and we will try to answer them through our portal or some other channel. Okay, so that's it for today's session. Keep following us on YouTube, Instagram, and Facebook, and try our app. There are many things that are more helpful in making you prepare for your competitive exams. Just try our app. So until next session, take care. Bye.